Hello, everyone, and thank you for joining today. I am Mendelevich from Altius Institute of Biomedical Sciences, and in the next 20 minutes, I will aim to convince you that current common practices in allele-specific expression analysis produce a lot of false positives, and will show you that this can be fixed with almost no extra cost when using RNA spike-ins and appropriate processing of the data. The work I will present here was published in the special conference issue. The respective R package control freak is available at GitHub. The overview of this talk is present on this slide. I would first define allele-specific balance in transcription, then we would discuss the presence of technical noise, which is important to account for, and finally, we will discuss our new approach that allows us to perform both accurate and cost-efficient allele-specific expression analysis. In short, that means that no sacrifices are required anymore. First, let me define allelic imbalance. In diploid cells, genes are present in two copies, one maternal and one paternal, and often shows unequal expression from parent alleles. Transcription of each allele is an integral output of the gene regulatory machinery. Since both alleles are in the same nucleus, they share transregulatory environment, so allelic imbalance very cleanly isolates this regulatory signal. Summing up, analysis of allelic imbalance is an effective tool to understand genetic and epigenetic mechanisms that regulate gene expression. Even though the most interesting question is often how allelic imbalance correlates with phenotype, we need a way to measure the signal and deal with statistical questions first. We aim to measure relative proportion of transcription levels from both alleles in the live cells, and then ask one of two main questions. Is it different from some point estimate, for example, balanced 50-50, or is it different between two different conditions? Since the initial proportion is unknown, we rely on observations. We are estimated allelic imbalance from reads produced by sequencing, and then allelic will result according to distinguishing variance. Allelic imbalance is computed as a proportion of transcription level of one given allele, and can take any value from 0, which is complete paternal bias, to 1, complete maternal. And the questions about differences may be now formulated in terms of statistical significance. It's common knowledge that gene coverage determines the wideness of distributions, and thus our statistical confidence in the observed signal. So do the experiment-specific noise, that without proper accounting may lead to an expected false positive rate. Here we come to the second part, where we will discuss origins of technical noise, the ways to capture it and account for it in allele-specific expression analysis. Variability between two samples depends on the point of division. Two in silico sampled batches from the same sequenced library will be less different from each other than separately processed libraries from the same RNA. And even more diversity will be present in biological replication. In many analysis types, it's extremely important to distinguish between those biological and technical noise components due to their difference in their nature. And also biological variation may be of some interest itself, when technical variation bears a pure artificial noise of a measuring procedure. The binomial model of allelic imbalance represents a situation of data sampling without additional variation present in the data. Although there is many evidence present in the literature that binomial assumptions are often over-optimistic, it's still widely used. But its usage results in high false positive rate in allele-specific expression analysis. If we will produce lots of technical replicates from the same RNA, we will clearly see the overconfidence of the binomial model. This figure shows the genes that were confidently determined as imbalanced, but towards opposite alleles in different technical replicates. Moreover, the variability of respective false positive rate will depend on the choice of experimental protocol. Difference between assumed variance from prior model and observed from the data is called over-dispersion. In our case, the prior model is binomial. If we will account for dispersion in our tests, that will return us the adequate level of confidence in our observations. How one may measure technical dispersion? We can consider multiple measurements and compare the results. This may be done having technical replicates, aliquots of the same RNA which were processed in parallel. In our previous work, we have shown that our dispersion in poly-A RNA-seq experiments behave like widening on experiment-specific quality correction coefficient, which is independent from coverage. The intuition is like that. We should consider and treat the observed gene coverages as they were q times smaller. Summing up, the problem of accounting for dispersion in allele result data may be solved when having replicates. When our dispersion is calculated for each sample and accounted for, the accurate differential analysis for allele-specific expression becomes possible. The only problem is that library preparation is a costly process, and if it's required to have 2 or 3 replicates per sample, then there might occur a desire to sacrifice statistical accuracy in favor of producing more experiments. Thus, we would ideally want to lower the expenses, so no more sacrifice is needed anymore. The natural idea was to add RNA from foreign sample to all main samples in experiment just before library preparation. Since both components in a mix undergo the same experimental processing, they should accumulate equal low dispersion. So, spiking components for all the samples may be used further for dispersion estimation, and the computed over dispersion measures may be assigned to each of main samples, which doesn't have technical replicates themselves. And right ahead, that worked. But first, we need to introduce some changes to our previous analysis. Since now we do not expect all the samples to have similar over dispersion, and we want to move from pairwise comparisons to utilization of information from the whole bunch of samples. And again, one of the most important things here is that now the additional experimental costs compared to protocol with one technical replicate consist of one extra RNA preparation for RNA spiking and some extra sequencing depth. In the next couple of slides, I will describe the algorithm of the or dispersion measurement. Let us first introduce the main result. That proposed experimental design and computation methodology works. We've produced various data testing different mixes, organisms, and experimental conditions, and having technical replication too. The figure shows that measured or dispersion is highly correlated for two components in an RNA mixture, so IQCC measures computed on spiking components may be fairly used to account for or dispersion in main samples. Now about the procedure. Since the main advantage of spiking methodology is the number of samples to which spiking RNA was added, we want to aggregate all information available from spiking components. To do so, we will feed n plus m parameters, where n is the number of genes and correspond to allelic imbalance values, and m is the number of samples or individual QCC values to feed. We want to iterate estimating allelic imbalance values and fitting IQCC for each sample until convergence. Note that IQCC are fitted independently in each fitting cycle here, so it could be easily parallelized. But one problem lies in estimating of allelic imbalance values. Usage of allelic imbalance estimate computed using all samples excluding the particular sample G when fitting its QCC value will result in overestimation of it. The reason is simple. Used allelic imbalance value itself belongs to some distribution around some true proportion. In opposite, including sample G in allelic imbalance estimation will result in underestimation of IQCC. Among other problems that we'll discuss later, that means that sometimes the fitted IQCC is smaller than 1. Since beta binomial family doesn't allow under-dispersion, we cannot use it. In our method control freak, we implemented modified Polya-Urn model, which in addition to regular beta binomial parameters alpha and beta, have a parameter D, which works like a switch between over and under-dispersion compared to binomial expectations. The intuition for this model is like that. You have an urn of colored balls, where colors represent transcribed alleles. You take a ball from it, write that down, then returns D balls from the same type back. The number might be 0. Then it will be corresponding to binomial, or sampling with return. Then positive D corresponds to over-dispersion and negative to under-dispersion. This model allows us to perform a tweak in case of small number of samples, which is the usual situation for technical replication. When we have lots of samples and each of them has a small relative library size, the upper estimate will be close to precise one, and no additional steps are needed. And we're working with technical replicates. The precise estimate may be computed as geometrical mean of under and overestimated IQCC values. Finally, a few more slides about robustness of the methodology. First, there are only 3 recommendations of the choice of spiking sample. It obviously should be able to undergo the same library preparation process as the main sample. Second, the organism should be well annotated, and having more frequent polymorphism will allow you to be more efficient and keep more reads as allelic resolved. Third, it should be distinguishable from the main organism, but note that even mouse and human almost do not cross a line on each other. Moreover, it is possible to simulate diploid organism with a mix of 2 strains. We have also shown that it's not necessary to keep the proportion exactly 50-50. You do not need to add a lot of spiking RNA. You need hundreds of thousands allelic reads for making an accurate IQCC estimate. That means that if total library size is sufficient enough, then spiking proportion may be relatively small. Finally, our methodology is very robust. It's enough to have only several hundreds of genes, and it doesn't depend on gene coverages, and it may operate with small amount of replicates. The take-home message is that proper measuring of accounting for technical noise enables accurate differential allele-specific expression analysis. Technical noise can be measured and accounted for when using either spiking RNA or technical replication, and we developed a computational tool for that. It is named Control Freak and available at GitHub. I thank my co-authors, and I also thank ISCB for partial funding of my conference expenses and to Grads for supporting the work. Thank you for your attention.